This is the first of 4 video segments in which we will be discussing active propagation of action potentials in dendrites. Before we begin our discussion of active propagation in dendrites, I'd like to give you one more example of cells that display spontaneous oscillations and bursty behavior. And these are the thalamocortical relay neurons that are found in thalamic nuclei. The job of these neurons is to receive sensory input from peripheral sense organs and then relay this input to the sensory cortex. These cells are found in one of 2 physiological states. In awake animals, these cells function to faithfully relay this sensory information to the cortex. In this state, the cells are depolarized, and the depolarization arises from input from brainstem nuclei. There's a serotonergic input from raphe nuclei neurons and a norepinephrine input from locus coeruleus neurons. And in this state, the sensory information arriving in the thalamus is faithfully relayed to the cortex. However, these neurons can also be in a second state. In sleeping animals, these relay neurons fail to relay sensory information from the periphery to the cortex. In this state, the cells are hyperpolarized. The excitatory input from the brainstem nuclei is halted, so the cell's hyperpolarized, and in this state the cells show a natural oscillation, and superimposed on this oscillation of membrane potential is cell bursting. When the cell's depolarized, the cells burst and send this bursting signals to the sensory cortex. The oscillations in these thalamocortical relay neurons arise from several different physiological mechanisms. First, the voltage-dependent channels, the voltage-gated channels in these cells lead to an intrinsic oscillation of the relay neurons, just as we saw for the inferior olive neurons. In addition, there are reciprocal connections between these thalamocortical relay neurons and neurons in the thalamic reticular nucleus, and these reciprocal connections also lead to oscillations. These oscillations, in fact, as they are relayed through the bursty behavior to the cortex, are responsible for the slow wave EEG patterns that are seen in sleeping animals. The diagram at the bottom shows the 2 physiological states of thalamocortical relay neurons when cells are hyperpolarized and they have a resting membrane potential of about minus 65 millivolt. We see this oscillation, and at the peak of the oscillation there would be several spikes. There would be a burst of spikes at the peak of each of these oscillations. However, if the thalamocortical relay neuron is depolarized, and in this case it was depolarized experimentally by injecting current into the cell, and this raised the membrane potential from minus 65 millivolts to minus 58 millivolts, then the oscillations cease and the cells spike, and if these cells were receiving sensory input, the sensory input would be faithfully relayed to the cortex. I've reproduced the voltage recording from the thalamocortical relay neuron that we saw in the last figure, and as can be seen, the neuron switches state from a hyperpolarized mode when the oscillations are seen to a tonic firing mode when the cell is depolarized. And I'd like you to think of why this switch in behavior occurs. Think in terms of the voltage-gated channels that generate the oscillations, and based on what you know about these channels, why would depolarization lead to a switch in the state of the cell from oscillation to tonic firing? We'll discuss this question in class.